We begin our study of animal diversity looking at the group of animals that is considered to be the basal taxon. That is a group of animals that is believed to have diverged earlier from all the other lineages of animals we have in the animal kingdom. And that is going to be the phylum Porifera. The word porifera literally means covered with pores. And there are two kinds of pores you can recognize in the body of a sponge. One is going to be the smaller openings through which water goes into the body of the sponge. Those are going to be known as ostia. And there's going to be, in most sponges, one larger opening that is going to be known as the osculum. And through that osculum, water is going to go out. Depending on the size of the sponge or depending on the complexity of the sponge, some sponges may actually have multiple osculi where water will be going out. This is going to be a real primitive type of animal in the sense of not having tissues. And by tissues, we mean a special type of grouping of cells of a similar kind with a similar function. And there is a degree of collaboration between those cells and also certain type of cellular junctions you may recall learning about when we studied about cells, some of those tight junctions that help fluid from moving between cells, or anchoring junctions that keep cells together. That level of cell junction is not seen in the cells that make up the body of a sponge. So without tissues, obviously there's going to be no presence of organs either. Even though sponges don't have any tissues, sponges are going to have different types of cells, which can be seen in the form of layers. For example, one type of cell that is really important for the life of a sponge is going to be the choanocyte. Now I'm going to enlarge my presentation so that I can show you a little better what these cells look like. Here's a choanocyte. It's kind of like, looks like sideways, but some people describe this like a flower vase, like something like a cylinder. And you can see here that rounded bottom, surrounded by a collar portion of the cell through which a flagellum is going to be projecting out. The collar produces a sticky material that little food particles can be trapped with. Those food particles are going to travel to the base of the collar, and then the plasma membrane on this part of the choanocyte will engulf those food particles by that process of endocytosis, which means cell eating, that is known as phagocytosis. So by phagocytosis, food particles are being brought in, and then there's going to be some partial digestion taking place inside of the choanocyte. But notice that food vesicles can also be shared with another type of cell you see here, which is called the amoebocyte. These cells get their name from the pseudopodia, these cytoplasmic extensions that can work like a foot or like a leg for crawling on surfaces. And so that is going to be the two of the types of cells we can find in a sponge. We have the choanocytes, which form a layer lining the internal compartment of the sponge. There's going to be these amoebocytes, also known as archaeocytes. The amoebocytes, in addition to helping with digestion, they also help distribute nutrients to other parts of the sponge. And also they have the potential of working like a stem cell. The amoebocytes can become any other type of cell in the body of a sponge. A third, also important type of cell you can find in sponges is going to be the pinacocytes. The pinacocytes form the external covering of the sponge, kind of like the skin of the sponge. I don't like to use the word skin because it kind of gives evidence of tissues, and remember that sponges don't have tissues. But the sponges do have this outer covering. It is made of flattened cells that look like little scales, and that layer of cells is made up by pinacocytes. In addition to those layers of cells, another important structure that is worth talking about in terms of the sponge body is going to be the spongocele. This word you see right here. Spongo means sponge, and seal means cavity, like the main cavity in the body of a sponge. This cavity is not of any embryonic origin. It's just simply a place where water collects before it goes out through the larger opening, which is known as the osculum. The sponge is going to be an interesting animal because they also have an endoskeleton. Endo simply means internal. Many times students come to my class thinking that the only animals with an endoskeleton are the vertebrates with their vertebrae made of bones, but they're not the only animals with endoskeleton. Remember that endoskeleton doesn't mean bone, simply means an internal skeleton. And the sponges is going to be one of those animals with an endoskeleton. If we can magnify this image again, we can find evidence of that endoskeleton. And this sponge is going to be made in the form of sharp, spiny objects known as spicules. These spicules are secreted from materials. I'm sorry, the spicules are made of materials that are secreted from the amoebocytes. And in some sponges, the spicules are going to be made of calcium carbonate, and sometimes they're going to be made of silicon dioxide, the same material you find in glass. And in some sponges, the endoskeleton is actually made of protein, a protein known as spongin. And those are ways in which we can actually classify sponges. The main criterion for the classification of sponges is the material of the endoskeleton, the material of the spicules or the spongin that makes up the internal support for the animal. There are three important sponge classes that are worth mentioning. The sponges of class Calcarea have spicules made of calcium carbonate. We call them also the calcareous sponges. The members of class Demospongia are going to include the bath sponges. Some bath sponges are going to be used commercially for the purpose of cleaning your skin. If you go to a place like Bath and Body Works, you may find natural ocean sponges that are used for cleansing your body because they are believed to be made of this protein that is not going to abrade, it's not going to harm your skin. Some members of Demospongia are not used for bath purposes. They actually have spicules made of glass, of silicon dioxide, and you don't want to use those sponges for washing your skin. There's also going to be another class of sponges, a class of sponges known as Hexactinellida, and the name of the class has to do with the nature of the spicules. So the spicules in the class sponges, Hexactinellids, is made of silicon dioxide like glass, but the spicules are characteristically, many of the spicules, not all of the spicules in all the species, but many spicules have 6 points. So hexa means 6. Hexactinellid sponges often have spicules with 6 spikes or 6 points. And so these are three of the classes we can recognize in the Phylum Periphera. And there's going to be more details about the sponges you will be learning about in lab. In addition to sponges, in this video, I want to introduce another group of animals that are relatively simple. Now, these are going to be part of the clade known as a Eumetazoan, meaning animals that are going to have true tissues. And so within the Eumetazoans, the most simple animals are going to be the ones that have radial symmetry. Previously, we talked about sponges, a different phylum of animals. The sponges have no symmetry, which we call asymmetrical, sometimes radial symmetry, but that's hard to see. With the phylum Cnidaria, we actually find a well-defined radial symmetry. And that simply means, as you recall, that the body of the animal can be cut exactly in half and produce equivalent mirror images. For the phylum Cnidaria, we are going to see animals that have a gelatinous body. There's going to be an external layer of cells that is known as the ectodermis. And the ectodermis actually, this differentiates into a type of a skin layer, which sometimes it's also described as the epidermis. There's going to be another internal layer forming a sac inside of the animal. And this sac or pouch is surrounded by cells of the gastrodermis. If you remember embryonic development from the previous chapter, there are some animals that are called diploblastic because during embryonic development, they form only two layers of embryonic tissues. There's an ectodermis, there's going to be an endodermis. So the ectodermis in the sea jellies develops into the epidermis, and the endodermis develops into the gastrodermis. There's also going to be a gelatinous layer known as the mesoglia, and that is how the body of these animals is usually going to be designed in terms of layers of cells. The cavity inside, or the space inside of a cnidarian, is going to be known as the gastrovascular cavity. Here we have a compound word. Gastro means digestion, and vascular means circulation. When food is brought into the gastrovascular cavity of a cnidarian, it is going to be partially digested, and then those partially digested nutrients can be circulated into the extensions of that gastrovascular cavity that can reach far into the tentacles of the body of the animal, or anywhere here within the main central region of the animal itself. So that is going to be one important detail about cnidarians I want you to remember: the layers of cells, the gastrovascular cavity, and the function of this gastrovascular cavity. We mentioned the radial symmetry. One detail about these animals is that they have an incomplete or closed digestive system. So food comes into the animal through the mouth. In this animal over here, the mouth is going to be right there, always surrounded by tentacles. And so food goes into the mouth, and when materials that are not digested need to be expelled, they're going to go back out through the mouth. Same here. Food can be partially digested. Materials not wanted by the animal will be excreted through the mouth. And so that's a closed digestive system. There's only one opening. This may not sound glamorous, what I'm about to say, but it's true. For animals with a closed digestive system like cnidarians, the mouth and the anus is exactly the same opening. So that's how food goes in and how wastes are going to be excreted. The mouth is also, I mentioned, it's going to be surrounded by tentacles, and tentacles are going to be the places where you find these stinging cells called cnidocytes. The word cnidocyte is what gives the origin to the word cnidaria. But what is the meaning of cnido? Cnido comes from a Latin root that means nettle, means like a stinging type of an object. And that is what animals use for capturing prey. The cnidocyte cells have inside a stinging capsule known as the nematocyst. And when that nematocyst is fired, it can deliver a venom that can paralyze prey or produce pain. That makes it easier for the cnidarian to then ingest the prey. Or the nematocysts can be fired and then used as defense to inflict pain on an animal that could be a threat. We mentioned the gastrovascular cavity. We mentioned already that these animals are diploblastic, meaning they originate from embryos that have only two layers of cells, ectoderm, endoderm. They have a skeleton. All animals have some form of a skeleton. In this case, it's a special type of skeleton known as a hydrostatic skeleton. And the way hydrostatic skeleton works is by regulating water pressure, hydro, and that happens when water moves into the gastrovascular cavity, filling up the cavity. Then contractions of this animal induced by fibers, contractile fibers and contractile cells in the mesoglia can put pressure on that water so that it is ejected out through the mouth, and something like a Medusa can float away in that direction. Or in the case of an anemone, when water is expelled, they can bring the tentacles in And some anemones actually can move from one location to another uh, using the help of this hydrostatic skeleton. So there are three things that I want you to remember about hydrostatic skeleton. One is that it requires water pressure. This water pressure is going to be regulated by the help of a contractile uh, cells and in contractile fibers. And there's also going to be a need for a container, kind of like a sac, like a tissue. So water, contractile cells, and a tissue that is going to be forming the cavity where water goes in. These are the animals with the simplest type of nerve uh, cell arrangement. It is described as a nerve net. And this nerve net is useful for like regulating the hydrostatic skeleton, pushing water out, bringing water in, and for some cnidarians to go from one place to another. Before moving on to my next slide, I just want to point out that there are two main body types in cnidarians. There is the cylindrical body type known as the polyp. Polyps are, for the most part, just settled, attached to one place. But I mentioned just a moment ago that some anemones can actually detach from the substrate where they live and change locations. And sometimes they do this to avoid a predator or to move away from another anemone that is causing too much competition. There is another type of body, and that is going to be the Medusa type, and that is shaped like a bell with the mouth dangling down towards the bottom of the ocean, and the tentacles are also going to be dangling down. That is going to be helpful when we look at the main classes of cnidarians we want to remember for this class. One class is going to be the Hydrozoans, a second is Zyphozoa, and a third is going to be Cubozoan. These three first classes are also known or described in your textbook as the Medusozoans because you can see the Medusa body type here. The hydrozoans can live in fresh water. There's a few that live in rivers or streams or ponds. Some of the members of this phylum are also going to exhibit Medusa and polyp stages. Examples you will see in lab include Hydra and Obelia, and I will save the observations and discussions about these two representatives for your activities in lab. The Zyphozoa is going to include most of the sea jellies, what people call jellyfish. It's kind of like a name that is not the best name for the sea jellies because they're not fish. So sea jellies is better. So animals with this Medusa type of body that you can find in the oceans, that is what is classified into this class, Zyphozoa. And the Cubozoa, which you can see also pictured in this illustration, have a body that is shaped like a cube, and for that reason they get the name box jellies. There's a species that lives in Australia, the oceans in Australia, and is one of the most venomous animals known in this planet. Then we have one more class, and that is going to be the Anthozoa. And these are going to be the anemones, also corals. Animals that form the coral reefs in the ocean are going to be there. There are other very interesting looking animals known as sea fans and sea pens. Those are also cnidarians, members of the class Anthozoa. By the way, the word antho means flower, and zoa means animal. Now, flowers are characteristic structures of plants, some plants, and this is not a plant, make no mistake, this is an animal. But when you look at an anemone, sometimes the tentacles kind of give the appearance like the petals of a flower. And so that's what the name, the appearance of the animal that inspired the name they have, the Anthozoans. That is all for the introduction to the cnidarians. And there's one more group that I want to briefly mention that is going to be the phylum Ctenophora, or the comb jellies. These animals have 8 combs of cilia, and they have biradial symmetry, meaning that the body can only be cut exactly in half in 2 different ways. These cilia combs are going to be characteristics of the animal. These are an important part of the plankton of the oceans that other animals are feeding on. They are predators themselves. They actually can discharge these as sticky tentacles for capturing prey. They also have a gastrovascular cavity. They have two main tissue layers and hydrostatic skeleton, like the one we just previously described in the phylum Cnidaria. That is all for this introduction to the basal animals, the sponges, and for the simplest of the eumetazoans, the members of phylum Cnidaria and Ctenophora.